Okay, so we just went through how we go from pyruvate, which is a 3-carbon structure, to your acetylated acetyl coenzyme A. And then this is 2 carbons that we're interested in. Obviously, there are more carbons in the coenzyme A, but 2 that we're interested in. Now we're going to take those acetyl-CoA groups and put them into the citric acid cycle. It's the same thing that we did in glycolysis. I'll hit the big picture in terms of what's going on in each step. I might pick a couple to look at the mechanisms, but for the most part, we're going to go light on the mechanisms and stick to the global overview. And if you look at so this is standard delta G here. What's going on? The first step is highly exergonic, right? Because there's a lot of energy in that thioester group. So there's an exergonic reaction. Couple of others here. Anytime there's a decarboxylation that's occurring, we have exergonic reaction. But we also have some endergonic reactions, right? This one is pretty low, so not much going on there. But we have some endergonic reactions, and again, particularly for this last step, positive 29 kilojoules per mole, that's a lot. So we aren't going to couple this with an exergonic reaction, so we need some mechanism by which we can drive this forward, and we do that by manipulating the actual delta G by keeping the concentration of oxaloacetate relatively low. So this is a citric acid cycle. I'll go through in a nutshell here. We have our acetyl-CoA group. It links up with the 1, 2, 3, 4 carbons of oxaloacetate to form the 1, 2, 3, 4, 5, 6 carbon citrate. We will isomerize that citrate using the enzyme aconitate. So we're actually going to add a water to it and then take it away. But the end result here is that we move this hydroxyl from this carbon over to this one. This actually forms a better leaving group. If you look, this is let's see, this carbon is attached to 3 other carbons. So that's a tertiary alcohol, is that right? Whereas this carbon is only attached to 2. So that's a secondary one. You're going to test me on my organic chemistry. And then one of these has a little bit better chemistry going on. I'll leave it to you to figure out why a secondary alcohol would be better than a tertiary alcohol for some good chemistry going on. Y'all should be learning that in organic chemistry, maybe as we speak. All right, so we form this isocitrate. And then we are going to remove a carbon dioxide. And if you look, zoom in here a little bit, at this, I'll erase this, at this citrate, carbon dioxide there, carbon dioxide there, carbon dioxide there. We're going to remove one, and kind of looking at the structure, it's just a matter of picking one to remove. Obviously, the chemistry is going to kind of dictate that, but you have a 1 in 3 chance of getting it right, just saying we're going to go through a round of decarboxylation. So the enzyme isocitrate dehydrogenase does that, right? It takes isocitrate, and it's removing some electrons and then process of carbon dioxide there. So we remove, in this case, this carbon dioxide, which one? This one that was here. You form a 5-carbon structure, 1, 2, 3, 4, 5, alpha ketoglutarate. And then in the process, every time you create a carbon dioxide, you're going to make an NADH. So in the process, you generate an NADH, and then you're going to do it again. We're going to remove a carbon dioxide. We are going to generate a molecule of NADH. And then we are going to form a 4-carbon structure, a succinyl group, and then we're going to attach that to coenzyme A. And this is the same coenzyme A that we saw before. Not literally the same, but the same molecule. So we're going to form a succinyl-CoA. And then very similar to pyruvate oxidation, we're going to the alpha-ketoglutarate dehydrogenase complex is very similar to pyruvate oxidation in terms of forming the succinyl-CoA. So then we're going to hydrolyze the succinyl-CoA, get rid of the coenzyme A, and that process create ATP, and then form a free succinyl group. And again, now that we have this 4-carbon structure, all we're going to do is oxidize it to get back to oxaloacetate. And if you just look here, 1, 2, 3, 4 carbons, 4 hydrogens, there's a 1-to-1 carbon-hydrogen ratio in succinate, and then it is a 2-to-1 carbon-to-hydrogen ratio in oxaloacetate, right? So you can see where those electrons are coming from. So first time we dehydrogenated, we form a FADH2. The enzyme that does that is succinate dehydrogenase, kind of easy, right? Substrate is succinate, the activity is dehydrogenase. We will see this is complex 2 of the electron transport chain. So this feeds directly into electron transport chain. We're going to form a fumarate here. Next thing is to add a water across that fumarate to form malate. And then we will take that malate and dehydrogenate it again to form oxaloacetate. The enzyme that does that is malate dehydrogenase. And then this oxaloacetate is ready to pick up another acetyl group. And there we have it. This is the citric acid cycle. 8 steps, 8 enzymes, plus a aconitase, and 8 intermediates that take us through to the end. And so again, we'll go through, I'll pick out some of the things that are interesting here, but we'll try and go through this relatively quickly. Have oxaloacetate, 1, 2, 3, 4 carbons. We have 2 carbons from the acetyl group. The enzyme that catalyzes that is citrate synthase, right? So we're making a citrate here from these 2 guys, and you're forming a 1, 2, 3, 4, 5, 6 carbon citrate molecule. A aconitase is going to add a water and then remove a water to move this hydroxyl group. So you're going to go from citrate to isocitrate, and then this isocitrate is then going to be decarboxylated to form your first NADH. So we're going to move some hydrogens to form that NADH, and then to form a 5-carbon alpha-ketoglutarate. So 1, 2, 3, 4, 5 here, and we lost 1 carbon dioxide. So if you're counting, we lost carbon dioxide from pyruvate oxidation, and since there are 2 pyruvates for every glucose, we lost 2 carbon dioxides there. And then now we've lost 2 carbon dioxides from isocitrate, and so we have 2 more to lose after alpha-ketoglutarate. Okay, so we have this 5-carbon alpha-ketoglutarate, 1, 2, 3, 4, 5. Again, this complex looks like the pyruvate oxidation, TPP, coenzyme A, lipolysine, NAD, FAD. So very similar, except that the substrate here is alpha-ketoglutarate and not pyruvate, right? The end result is that you remove a carbon dioxide and you form a 4-carbon structure, 1, 2, 3, 4, attached to a coenzyme A. And then the succinyl-CoA, you remove it, and it's, again, a similar mechanism. You're preserving a lot of the energy in this thioester bond here. So hydrolyzing that thioester bond gets the energy. That's enough energy to make the production of GTP from GDP, an inorganic phosphate. You release that coenzyme A, and then you're left with a free succinyl group there. And then we're going to oxidize the succinyl group, so transferring electrons from succinate to FADH2. So we're reducing FAD to form FADH2, and then you have this fumarate, and this fumarate is a lower, higher oxidation state than the succinate that preceded it. So we're taking those high energy electrons out of there. The next step is to add a water across, and this occurs in a stereospecific way that Dr. Young, I'm sure, will go deep into. But for our purposes, adding a water across this bond forms a malate. And then this malate allows us to form oxaloacetate in the dehydrogenation process. And so we're going to take this malate, we're going to remove electrons from there and form our last NADH. And then we're going to go back to what we started with the oxaloacetate. So 4 carbons, 1 to 3, 4 carbons. You've lost, you're much more oxidized than you were when you started with succinate, right? You removed these electrons associated with that carbon right there. And we are ready now to pick up another acetyl group and begin the cycle all over again. So the result here, 1, 2, 3 NADHs, 1 FADH2, 1 ATP, 2 carbon dioxides, and this whole thing is times 2 because we generate 2 pyruvate for every molecule of glucose. And again, in terms of regulation, just like in glycolysis, you're going to have certain points of regulation, and these will be negatively affected by indications that the cell is at rest, so inhibited by ATP, by NADH, acetyl-CoA, right? Inhibited NADH, ATP, and activated by ADP, NAD+, so forth, and so on. So that should be intuitive at this point. Activated by things that show that the cell is at rest. Inhibited by things that show the cell is at rest. Activated by things that show the cell is at work. And we won't talk about anabolic pathways, but there are ways to get, and I think I mentioned this before, from the citric acid cycle, we can get into all the other biological macromolecules that occur. When you do that, so amino acids, heme, fats, when you do that, the first thing that happens is that the cell restores the steady state concentrations of the citric acid cycle. So it gets back to where it was, and then you can go back into energy production. And so that's what these analyphoretic reactions are that we won't talk about. And here, again, we would go into detail on this on an advanced biochemistry course. Oh, good. And that is it. So now, what have we done? We have, just use this clicker question because I need some white space here. So we have broken down pyruvate. Pyruvate doesn't exist anymore, and we have gotten rid of all of the atoms of the original glucose. In the form of carbon dioxide, and then the hydrogen atoms, which have all sorts of uses. We have generated in this process 3 NADH in the citric acid cycle plus 1 NADH in pyruvate oxidation, and this is times 2 because there are 2 pyruvates. So here's 6 NADH plus 2 NADH. We've generated 2 times FADH2 in the citric acid cycle and 2 times ATP in the citric acid cycle. And just remember, we also generate 2 ATP. So all of this is TCA and pyruvate oxidation. And then we generate 2 ATP and 2 NADH from glycolysis. Oops. So in total, we have, what is that? Two, change the color here. Let me go down here. So in total now, we have 4 ATP, and this can be used directly by the cell. And we have, what is this? 2 plus 8. Oh, wait. No. 8 plus 2. 10 NADH. That has lots of energy that we need to get. Two FADH2. That has lots of energy that we need to crack to get. And then 6 carbon dioxide that has no energy, so we need to get rid of it via our exhalations, right? And then that's it. And so the next chapter is to look at how to get ATP out of these reduced electron carriers that we've made. And we'll do that via the process of oxidative phosphorylation. All right.